All right, good evening. I started doing something 5 years ago, and we are close to the 5 year anniversary of when COVID-19 first came to the US. And of course, we had the spring of 2020, which no one needs help remembering that that was very different of a time. When we were all locked down, not going out, I wanted to try to use my immunology training as a structural immunologist with a distinct perspective on this whole thing to talk about how COVID looked to me. But I always wanted to base it on the data, so I called it a data update. Now we're in February 2025, 5 years later. And now I want to say, okay, now that we're out of the thick of things, it's hard to tell what's going on in the thick of things. Science requires replication. It requires multiple people looking at the same thing. It requires peer review, and it requires admitting when you're wrong. And I've done some of that myself. But I do want to look back at COVID-19, and I want to say one really good, solid scientific analysis of a number that I was worried about throughout the entire 5 years ago period that we were talking about. Finally, we got a good paper that I really trust as far as their analysis goes. So let's talk about that paper. And I want to return to that as sort of a 5 year anniversary of COVID. And hopefully this will be my last COVID-19 video ever, but I've learned to never say never. So I have this all on a website, and right now there's 69 videos. This one's going to be number 70, and it goes way back to, it says 4 years ago, because it's not quite 5 years ago, but it's almost 5 years ago. And I want you to see that this is involved with different audiences. I always tried to make these for a more general audience than say my journal clubs. It's not my classes. I tried to explain things, and it's always the way it looks to me. Again, I've made some mistakes, but I'm overall happy with how my immunology told me and gave me hope, actually, at sometimes about how things were going to go. And I want you to notice that there are some things on this that you see where I'm talking to my church. I'm talking to different levels of community. It's important for us to talk to each other about these things. This is my way of doing it. It's not meant to be the final word. None of these were ever meant to be the final word, but they're always meant to include a lot of data. And we want to be centered around the data because as flawed as data is, it's the thing that I want to look at. So in fact, on March 2, 2020, we were still in session for class, and I had a class session. This is from it right there. And this is the class session where I showed the SARS-CoV-2 COVID-19 causing genome. Right here, it's 4-point font, it's 30,000 characters, but this is the entire genetic material represented as A's, C's, G's, and T's. So the entire genome is right there, and I showed it to people before we even locked down. The good news is they were working on vaccines even at this point. And this is data. So I wanted to always base what I said on the data. Now back then we didn't know how it would go, but I said these are other coronaviruses. This is how a novel coronavirus, I think it will go. And one of the things I said is I think we'll be able to build immunity to this. There's a paper that just came out about how are the symptoms of it changing. You might remember back in the day, it was very hard to get a symptom list of exactly what the early symptoms of SARS-CoV-2 infection were. They seem to be all around, all over the place, because part of a novel virus is it presents in a novel way. And we really didn't have a good handle on it, and it also was provoking a lot of different effects in a lot of different people. But the thing is, as the population develops immunity against the virus, it sort of forces it down a certain road so it looks more and more normal. And we begin to have symptoms that will be, as the community evolves immunity to it, you'll be able to see that the same things happen. So what are these? This is in early 2025. Look at how the symptoms have changed over time in a scientific way. And so now it looks like, starting with Omicron, started to stabilize in that you could associate particular symptoms with SARS-CoV-2, increased cough, sore throat, runny nose, and sneezing. And you can talk about how other viruses have sort of done the same thing. It looks more and more like the common cold as we go on. It's taken years to get to this point. But one of the things I said is I don't know what the symptoms presentation is now. But I do know that our immune systems are very good at the job, and if we augment that with vaccines, then we can hopefully accelerate this timetable where we develop a defense against SARS-CoV-2. In the meantime, it causes a lot of damage, and it's a very scary thing, and it was a very scary time. You can't look at the virus the same way now as you did then. What I'm saying is right now, it has evolved to the point we have co-evolved with it. Our immune systems have developed with it, and the vaccine has helped, I'm convinced. And the Omicron strain was really the inflection point where it started to get more normal. And so exactly the way it started to get more normal is it's looking like other viruses, and it's having a normal set of symptoms going along with it. So I've jumped back and forth between 2020 and 2025. Now I want to go to the middle of those, March 2022. And that's back when X was still called Twitter, and I insist on still calling it Twitter. It's definitely changed. But I still find some good on it, believe it or not, from the characters that I don't find anywhere else. One of the characters is probably no longer on it, but in 2022, Siddhartha Mukherjee, who wrote a book that I had assigned to my class called The Emperor of All Maladies, and I actually assigned another book of his to another class of mine, a very good writer and also a physician. And just basically has a really good way of looking at things. And then on Twitter, Mukherjee posted, "Can someone please publish a homology map of all the beta coronaviruses that have been circulating? Because there could be some pre-existing immunity, because for all the novelty of SARS-CoV-2, it actually has 4 cousins that are common cold viruses that were circulating before. Now they're cousins, not siblings. And so the question is, if we have immunity to these particular common colds, and they're only about a quarter of all common colds put together. So there's spotty coverage, but is that coverage something that will help people?" And he was asking this question, and I jumped in and I said, I'm actually doing this with my students in class, where we are looking at the similarity of these cousin cold viruses to SARS-CoV-2. And based on the percentage of identity, which we can look at biochemically, we think that the immune response is about 1/3 the same at the T cell level, which is only one branch of the immune system. But still, that's something. That's something that suggests that cross-reactivity is maybe smaller than we might want, but it is real. And there is some defense that people have, even against this novel coronavirus, if they happen to have been exposed to the cold virus and develop T cells in the right way that will cross-react with the new virus. So the details of how we did that, we actually did it in an Excel sheet. It's not super fancy, and you can use online tools to predict which little snippets of the spike proteins, and actually this was little snippets of all the proteins in the genome, because remember, we have the genome. We basically took the genome, we put it into our tools and said, which parts of this will the immune system like? How much do they change when we're going from common cold to SARS-CoV-2? And basically, if things look the same, I colored them green. If things look different, I colored them red. And each of these columns, up and down, is one of those genomes. 4 common cold genomes, 1 SARS-CoV-2 genome. The more red you see, the more you don't have cross-reactivity. The more green you see, the more there's a possibility of cross-reactivity. You look up and down, and it's about, maybe 1/3 green. And again, this leads to our prediction that maybe 1/3 of the T cell activating parts of SARS-CoV-2 could have been pre-activated by one of the strains of common cold. I go into a lot more detail in this and other things. The cool thing about it is I was doing this in the undergraduate classroom, and part of it is what we did on computers because we couldn't meet in person. We were still able to research, and we came to a conclusion that I believe is part of the reason why we've been able to converge on these same symptoms for SARS-CoV-2 now, why our immune systems have been able to adapt, because some of them got a head start in being cross-reactive between common colds and SARS-CoV-2. Also notice this is not all green. Cross-reactivity is only a minority response. And so only 1/3 of the epitopes even have a chance, and it depends on your exact immune system whether one of these 1/3 will work with you or not. So, and this also fits with what we saw. I said this in 2022. It looks like that was the thing. Oh, and by the way, that was when I was explaining it in 2022, and it was actually happening. And I think I have the dates right here, so I should probably, yeah, this was actually in 2020 when the conversation started. This is my summary in 2022. In March 2021, Mukherjee posted this article in the New Yorker about why does the pandemic seem to be hitting some countries harder than others. He had relatives in India who did not seem to be hit as hard by SARS-CoV-2. And based on our conversation, he actually quoted me, he interviewed me, and I talked about how we did this project to measure the cross-reactivity, and we think that some of them come from common cold cross-reactivity. And so that was all, that was cool, you know, being, I'm usually looking for being cited in scientific journals. This is probably the first and only time I'll be cited in the New Yorker, but there we are, and there's my students who did the work on that I'm very proud of. The thing is, what happened after this, and this is a lesson about how hard it is to tell what's going on in the middle of things. Right after this was published, the Delta variant emerged, and it emerged in India first. And so this was a newly infectious version of the virus, and you can see the article was published right about in this lull right here. And this is SARS-CoV-2 infections in Delhi. And then you had this, right after we published that, about why wasn't India hit so hard, India was hit harder than ever by Delta. And so this made me wonder, in the thick of things, was I wrong about cross-reactivity? Because it looked like Delta was worse than the original. It looked like things were getting worse. And that's the thing I want to talk about. These are confirmed cases. There were a lot of deaths. But confirmed cases is only the easiest to measure thing. What really matters is infections. So we had a case fatality rate, and it was very high. And the question is, how big were the infections that were going on to have the infection fatality rate? What we knew is that independent of all that, the fatality rate for Delta was very high, and then it spread across, it came here, and it caused a wave in the US. And so it was bad, but how bad was it? At the time, being in the middle of it, it looked worse than it was, because I was worried, and I was also worried that I had just said the pandemic wasn't hitting India as hard because of this, and then it hit it harder. And but it's hard to see exactly what's going on with it, because the virus is definitely evolving and changing. It's outwitting some of the immune system, then it becomes more infectious. And so when I said it looked like it was getting worse, it looked like the case fatality rate and the infection fatality rate were getting worse for Delta than they were for Alpha. But then you have to say, well, were they really? In the middle of things, it's hard to tell. And that's where we have the perspective right now. So this is a study from 2021 that was looking at the Delta variant in particular. And you can see the transmissibility has increased by a large amount. The immune escape also increased. The good news is the transmissibility increased is mostly, if you had something that was more transmissible, it was less immune escaping. That's why you had the higher transmissibility increase on this graph in the lower right-hand corner of it. And yet you have lower immune escape there too. So this was one sign that the Delta felt really bad because it was so transmissible. But it was not really that much more immune escaping. In fact, it was more about its transmissibility than its immune escapability. But still, it was bad, and in the middle of it, you don't have time to say, how bad is this? You need to step back after years. And so I was a little bit like wondering, well, what's right? How is this virus? I said the virus, that we'd get more used to the virus over time. Why does the Delta wave look so bad? Well, one of the things that you can look at is you can look at the genomes themselves. So what I did with one genome, Trevor Bedford at the Fred Hutch Cancer Research Center did with all the genomes he could get a hold of. And he identified the different waves coming out and how the genomes were different. And he also connected that to how transmissible is this and how much does it avoid antibodies. There's two things the virus can do. It can transmit more quickly. It can avoid the immune systems that are already there. So we looked at all the different waves, and there are some waves that you never heard about because they never really were a big thing in the US of A. But the alpha wave, I remember hearing about that. The beta wave, I also remember hearing about that. And each of these has a transmission advantage above 1. That's why it becomes a wave, because it has a transmission advantage. The epsilon is the only one that has a transmission advantage around 1, and also, you probably don't remember an epsilon wave. Part of the reason is it was not as transmissible. The more you are to the right on this graph, the more transmissible you are. And so hopefully my head is not covering it up. Hopefully you can see there's a little bit of purple that the transmission advantage for delta was above 2 for the most part. Delta was highly transmissible, which is one of the things that you'd predict. You'd predict that it would be more transmissible because the virus has a pressure to become more transmissible. If it's more transmissible, it will survive more and infect more people. And this is one of the reasons why Delta looks so bad in the middle of it. Notice this is transmissibility and not fatality rate. This is whether you get infected, not whether you get hospitalized or in the ICU. And so this is another thing that an analysis of 6 million SARS-CoV-2 genomes. And what it does is it goes by each genome and the R value, which is a measure of the transmissibility of it. You start off with an R value of 1. You're in the lower left corner of that. And as you go along, you have the number of cases as the size of the bubble, and then you have the name of the viral genome that's been isolated. Now, we don't have the common names for these, like the Omicron, Beta, Alpha name for these, but I can tell you the big bubbles are the big waves that you remember. And I just want you to notice how this steps up over time. They become more and more infectious, and it's pretty much a common thing. And the first virus to get notably more infectious usually has a bigger bubble. You have the B117 bubble there. I believe that is alpha. And then the B16772 bubble above that, I believe that is delta. I'll have to go back and check. The one thing I know is way above my head here, you have the BA2, BA1.1, those are Omicron. And so those are much later. Those are coming about in, don't take my word for it. You should probably look that up to be sure. I'm pretty sure those are Omicron because that's around 2021, the December and things like that. And like I said, that seems to be a real inflection point, but it's becoming more and more transmissible. This is like an inexorable ratchet. But the good news is there's a limit to this. After a certain point, we began to catch up to the virus. So yeah, this is 1, 2, and 3 are the points that we're talking about here. And then we have Trevor Bedford, who looked at the viruses as branching out. Each different color is sort of a different wave. And what he has is he has date. Again, we always have a timeline where we have date going across. Going up on this case, he has number of mutations. So you can see that you're following this sort of steady line, but then you have a virus that jumps because it has a number of good mutations, and that lets it survive and transmit more. So you have this jump right here, and this is what created alpha. The big jump up to create that orange thing on the left is what created alpha. There's a similar one for beta for South Africa, but it's not as big of a jump, and that's because it's fewer mutations. And so what happened with Delta? Delta would outcompete the other variants, and unfortunately my head's sort of blocking, but behind my head there is a big, what you have is a timeline going across, then you have the alpha showing up in blue, and then it gets warmer and warmer as you move through the Greek alphabet. Delta is green. And I'm not sure if you can see it, but the green sort of overtakes everything else. This is the Delta wave, which overtook the world, especially in the fall of 2021, which is right about where my head is right now. So Trevor Bedford took these new waves and he said, how closely related are they to each other? So you have the original is about in the center of the graph. And then you have the different kind of waves branching out. You have alpha branching out to about the 8 o'clock position, beta and gamma branching out to about the 1 or 2 o'clock position. And then you have delta sort of doing its own thing. Delta was a big jump up in mutations and a big jump up in transmissibility. So it's definitely different. It's definitely worse in some way. But was it worse in fatality rate? That was my question. We could tell very quickly it was a big jump in mutations, and it looked like we were going backwards. It looked like we were losing the fight against the virus. But my immunology said we should eventually be able to catch up to this. And it shouldn't be that much as bad as it looks right here. The other thing I'd like to say is that Omicron was another big jump. It was in another direction. So it was down at the 4 o'clock position. But I just want you to notice the distances between the colors are a rough estimate of how similar these sequences were. Alpha, beta, and gamma were pretty similar. Delta was different in one way. Omicron was different in another way. This virus is mutating, it's knocking at the door. When it gets enough mutations to cause it to survive better, then it bursts out in a wave. And that's what we had happen at least 3 times with Alpha, Delta, and Omicron. So Omicron was a good surprise. I remember being worried about it when it came out, but there were some early signs, even back at the beginning, that this was actually not causing the fatality rate that we were worried about with the other ones. And so it soon swept over. You can see the Delta green is replaced by Omicron orange. That's shown in both the bottom and the top. And the top is sort of a branching tree. And you can see there's a wave of green on top around August 2021. But then around December 2021, there's a wave of orange. And the orange is the first Omicron. There's a second Omicron that came along, but there's really not much difference between them. Omicron was where the fatality rate was clearly much lower than it was before. So the question is, if I was thinking that fatality rate would get better over time, what's going on with Delta? I'm not surprised that it was more transmissible. I'm surprised that it looked like the fatality rate was going up when I didn't think it should. And it was very hard to measure. And also there's a lot of different things about the diverse populations it was getting into. It was worse for some populations, that's for sure. I'm just talking about the global view, which is the place in which I made predictions about the virus would get more transmissible, but it also would get less fatal. And so here's the severity that was shown up relative to other viruses. And this is in the midst of things. This is a 2022 way of looking at things. And what surprised me about this was that alpha, beta, gamma, and delta are sort of the same on this graph. Definitely Omicron's back down in severity. But alpha, beta, and gamma, are, at least beta, gamma, and delta, are similar. And if anything, the highest one is gamma or beta. This does not go with when I was looking at the severity and trying to estimate severity as we were going through. Like I said, delta was so much more transmissible, it looked more severe than it was. This is also an in-between analysis, but this is the first sign I had that, yeah, delta was bad, but it's not going up remarkably relative to gamma and beta at least. And it's not really going up that much relative to alpha. The error bars are still large, so I couldn't really, but this is the first, I couldn't really hang my hat on this. But still, it takes years to do good science on a global scale. Which brings us to this article that actually is far enough back to really do the analysis right. What is the case fatality rate and infection fatality rate for the worst years of the pandemic? And you always have to focus on something. They're focusing on Austria. Western European kind of nation and population. Very distinctive, and there may be differences with other nations. But this work is done well enough that I believe that most of it should be applicable to the entire globe. And here's the table of what they had, the overall CFR. So if you want to know what the CFR is, the population that they measured was 9 million, and there were 5.9 million infections, there were almost as many infections as population, and they 18,720 deaths. That was a 30-day mortality, a few thousand more if you go to 60-day mortality. So the question is, what does that end up being? Ends up being 0.34% 60-day mortality. And they break it down by age, and they break it down by vaccination level, whether they have hybrid immunity, which means that you have both been infected and have been vaccinated. And they even break it down by nursing home residency. So the overall IFR that they end up with is 0.16, or actually I should probably go with the 60-day, which my head might be blocking. It's 0.18 is the overall IFR that they have. So the infection fatality rate, and this is for the 2020 to 2023 region in Austria, is 0.18. And if you exclude nursing homes, it goes down to 0.13. But of course, it varied over time because the virus itself varied over time and people got more immunity over time. And what this shows is the CFR, which we are a little more certain about, and the IFR, which is more of an estimate, but they should go together. And on this study, I think they're both. The CFR is shown in the red line. The IFR is shown in a blue line. And you can see it's jumping all around on the left side of the graph. This is the original strain of the virus. Then you go through to Alpha, Delta, and then the Omicrons. You can see that the cases go up in the first wave. There's the 2020 wave where it really hit Austria for the first time. And then you have the Alpha wave, which did not hit Austria that much. Then you have the Delta wave. Then you have the Omicron wave, which was pretty much global. The Omicron wave is the biggest wave. And they were estimating what the undocumented cases were. You know, documented is where we have evidence, we have a test. And then they tried to estimate from that how many other people that didn't get the documentation of their infection, how many were infected. And in general, that's the green line. But you can see that they pretty much go together. The waves go up and down. The CFR and the IFR does pretty much drop from being really high, even up at around 1% in the first year of the pandemic, but then dropping to where it is, which is 1/10 of what it was now. Actually, let's put some numbers on it because it's hard to see exactly where it is now that we have such a low IFR. And remember, IFR is the number that we most want to think about. If I get infected, what is the fatality rate? The CFR is if I get tested, a positive test. Because we have fewer positive tests now, we have a lot of infections that don't get documented, don't get certified. So that number is, there's a gap between those two numbers again. But really, the graphs are not for everyone. What I want to say is what is the case fatality rate is what they're more certain about. You're not really estimating things. And this is actually an analysis from another account, and I want to give credit to that account at the bottom. This account put together this table, which I think is the best way of looking at things. So for the entire period, what is the case fatality rate from 2020 to 2023, including all cases and all fatalities during that time? Overall, if you had no vaccine or previous infection, the case fatality rate is 0.67% overall. So that's about a 1 in 200 chance of dying, which everyone would agree is bad. 1 out of 200 cases across all ages will become a fatality according to the Austrian analysis. Now, what brings it down, if you had previous infection, it brings it way down to 0.03%. The vaccine brings it down to 0.16, and vaccine plus infection is 0.05. Now, do not overanalyze the difference between those three columns. Because the vaccine was rolled out to the most vulnerable first, that means the vaccine numbers are going to be more inflated. More of the more vulnerable got the vaccine, and so you can't directly compare and say, oh, previous infection was 4 times better than vaccine. I'm not willing, even with this quality of data, I'm not willing to go there, but I'm willing to say it's all good. It was all working, and the age difference that we saw from the very beginning kept showing up. This is, but what I really want to ask is how does this is the overall average of all viruses, everything between 2020 and 2023. So the age was interesting enough. But what I really wanted to do is take the data, and I wanted to say, what is the case fatality rate when you go from strain to strain? The original wave had a case fatality rate of 2%. Again, 1 out of 50 people who were documented to have cases of it would actually die from it in Austria. Alpha variant, 0.8%. Delta variant, 0.5%. And Omicron, 0.08%. So the thing about Alpha and Delta, you see the case fatality rate is actually still going down with Delta. And that's what I was hoping that immunology said would happen. If we throw everything we've got at this virus, then we will bring the case fatality rate down. Delta appeared different, but that was an optical illusion because Delta was so much more transmissible. It ended up seeing more cases and seeing a lot more deaths just because you had so many more cases. So it's a bad thing. But the immunological rule that we can take care of this virus with patience and time and effort, and the extreme effort that was put into making the vaccines, and the extreme effort that was put into people caring for each other when they got sick, and allowing for each other to get sick and to take care of the stuff for them, that's all part of this. That's baked into these numbers. And so the virus was trying its best to outwit us, but we outlasted it at a great cost. There were many people who died that should not have. But the main thing is the overall idea that what we do against this matters and that we can beat this virus and it will get better over time. That's shown even for the case when in the middle of things I questioned it. And so that's what I want to say about this. That's the take home that I get from this. There's a lot of caveats and nuances that won't even fit into a video and definitely won't fit into a short form post like will go viral. But this is what I have for the best science. The case fatality rate did go down over time. And it was bad. A 2% case fatality rate is a bad thing. It was also age dependent. So we need to talk now about how will we look at data. And I want you to understand when you're in the middle of a crisis, social media is not a very balanced way to look at data. Even if you know the science and you're pretty certain that we'll be able to beat this virus eventually, I was a little worried during the Delta variant because it looked like I was wrong. But now looking at this, the reanalysis of the data says the general trend was right. And now let's research into the other things like long COVID and things like that. Let's find out, let's define what they are. And once we can define them as well as this study has defined CFR, if we can define long COVID this way, then I think we can get a handle on it and we can treat it and hopefully reverse it. So that is hopefully the last, looking back 5 years, there is a lot of things gone wrong. But at the very end, the immunological rule is that your immune system, plus our best efforts in other areas, do actually eventually beat this virus. And it is not something that is an unsurmountable problem for us as a society. But we need to look at the difference between how things look when you're in the middle of things and how things look when you're standing back at it after 5 years, with science to make you objective rather than social media to make you afraid. There are things to be afraid of, but science can be a balancing effort rather than a weapon to use. But it takes time. Okay, that's my interpretation and hopefully my final data update.